Hi guys, welcome back to my channel. It's Doc Junel. The subject at hand is Human Anatomy and Physiology, and the topic for this video is Cell Anatomy and Cell Physiology. Previously, we've already talked about the cells, and we said that the cells are the basic unit of life, which means that they are the smallest living things or the smallest units of life. And once cells of similar function come together, they then form tissues. In different types of tissues, when they come together, they form organs. And organs can form organ systems, and the combination of the different organ systems will then create the organism. So in order for us to understand the complexity of an organism, we must first begin with a very basic unit that created the organism, and that is the cell. Other functions of the cell, aside from being the basic unit of life, include cell metabolism and energy use. So we mentioned in our last video as well that ATP or energy is important to sustain life. And a lot of this ATP and energy are created actually in our cells. At the same time, a lot of this ATP that we produce are spent or used up by the cells as well. Moreover, we talked about organic molecules in our previous video, and proteins, lipids, carbohydrates, nucleic acids, they are all synthesized in our cells. It all begins there. Also, in order for stimulus, in order for the body to respond to different triggers, it's important that our cells are capable of communication or transferring the signals, transferring information from one location to another. Lastly, cells are important for the main purpose of reproduction, repair, and even inheritance. So these cells are able to function because they are made up of organelles. And organelles are the specialized units or structures that perform different functions in order to sustain the life of the cell. This includes your nucleus, mitochondria, ribosomes, and many other, which we will discuss in this video. All of these organelles, or most of these organelles, are actually located in the cytoplasm. The cytoplasm is the jelly-like substance that holds the organelles. It is the fluid that is found inside the cell. And the cytoplasm and the organelles are all kept in one compartment by the cell membrane, which is also called the plasma membrane. And this is the structure that encloses the cytoplasm. Basically, it acts as the wall of the cell. So let's begin with, of course, the very important structure called the cell membrane. So the cell membrane is actually this blue structure that you see here surrounding this entirety of the cell. It is represented by this blue wall. As mentioned, it is the cell wall. And it makes sure that the cytoplasm, which is represented by this yellow substance distributed all over the cell, it makes sure that the cytoplasm is kept inside the cell. And the cytoplasm is the one that holds the position of the different organelles. Now let's continue to talk about the cell membrane. The cell membrane or the plasma membrane is the outermost component of a cell. Basically, that's why it's called the cell wall, because it is the boundary between the materials found inside the cell and the materials found outside the cell. So the materials that are found inside the cell are called intracellular. That's quite easy to remember. Inside, so intracellular. And those substances found outside the cell is called extracellular. Extra meaning outside. So if we take a look, or if we compare the cell membrane to an object or a place, I would think of intramuros. So this is the top view of Intramuros. For those who do not know, Intramuros is called the walled city. Basically because Intramuros is a city that is bounded by a wall, separating it from the rest of the community. So let's imagine that the red lines that you see now is the cell membrane. So everything inside the cell membrane is called intracellular, and everything outside the cell membrane is called extracellular. So it is the cell membrane's job to keep all the substances inside secured from the substances found outside. Okay? Now, there's a very special characteristic of the cell membrane, and it is that it acts as a selective barrier. So, meaning it checks very well what substances can enter the cell and what substances can exit the cell. So we have here a good model, this one. This is called the fluid mosaic model. It is the globally accepted depiction of the cell membrane. So according to the fluid mosaic model, the cell membrane is composed of phospholipids, cholesterol, proteins, and carbohydrates. Basically, they are made up of organic molecules. But out of all these organic molecules, the main component of the cell membrane is the phospholipid. So the phospholipid, let's discuss it first, forms a bilayer, meaning it forms two layers of the cell membrane. And as we said in our last discussion, phospholipids has two regions, the polar and the nonpolar end. The polar being composed of phosphate and the nonpolar composed of the fatty acid. So the phospholipid is again a type of a lipid molecule. So lipids, as we know, are insoluble in water. Luckily, the phospholipid has two parts. This blue structure represents the phosphate containing region, and phosphate is polar, meaning it is soluble in water, and it is hydrophilic, meaning it loves water. And then this extension here, this is your fatty acid chain, which is non-polar, meaning it is insoluble in water, and is hydrophobic, which means it hates water. So what happens is the polar region is the one that is exposed to the water outside and inside the cell. So later I will show you that. So this is the phosphate end or the polar end that comes in contact with the water outside the cell and the water inside the cell. So the nonpolar region or the fatty acid chain, on the other hand, faces the interior of the membrane. So it is away from the water inside and the water outside the cell. So here is another or a bigger image of the fluid mosaic model. So I want you to notice first the top layer and the bottom layer. So actually, each of these two layers are composed of phospholipids. You can see the blue globular structures, those are all phosphate heads. Diba? And then at the bottom of it, you see the chains of fatty acid underneath. And then that is duplicated or mirrored underneath by again another layer of your phosphate head, and above is your fatty acid chain. So this top layer, it comes in contact with the water outside of the cell. Okay? So this is, let's imagine that this is the substance outside of the cell. So of course there's water there and other molecules and ions, no? Dun sa labas ng cell. So it is the phosphate part of the phospholipid na merong contact or nakadikit or nakikisalamuha doon sa water outside the cell. And then, underneath, on the second layer of your phospholipid, the phosphate heads also come in contact with the fluid inside the cell. Remember, your cytoplasm is a jelly-like substance which has water, of course. So it is the phosphate head also that comes in contact na na-expose dun sa water inside the cell. And the fatty acid chains are facing the interior. Both of them are facing the middle of the double layer or the bilayer. Moreover, we can see here several proteins in purple, in green, and even in red here. These are all proteins, okay? And the function of these proteins is they act as a bridge. Bridge for what? It acts as a bridge for different substances outside the cell to come in, and also the other way around. It acts as a bridge so that substances inside the cell can exit or go out of the cell. Moreover, we can also see here these yellow structures, which are your carbohydrate chains. So later we will find out what these carbohydrate chains are for. All right? So here is another image just to further drive the point, no? So the cell membrane is made up of two layers of phospholipid. That's the outer leaflet and the inner leaflet. So the outer leaflet has the phosphate end on top and the fatty acid chain at the bottom, while the inner leaflet has the phosphate head below and the fatty acid chain pointing upwards. Just to further discern the different structures, the circular ones or the globular ones are the phosphate or the polar heads, while the fatty acid chains are those two structures protruding from the phospholipid. Also, interspersed amongst the fatty acid chains are cholesterol. So cholesterol is again another type of lipid, and it has a very important role in the cell. Cholesterol provides cell fluidity. It provides the cell fluidity, meaning it allows the cell to be able to move. It allows the cell to be able to adjust its shape, no? Especially when there are many substances also surrounding it, no? Kumbaga, pag naiipit siya ng ibang mga structures, kaya yung cell kaya niyang mag-iba ng shape kasi meron siyang cholesterol. It gives the cell its fluidity. As mentioned earlier, we also have your proteins, and we have the first major type of protein here called integral proteins. What's special about integral proteins is that they penetrate the cell wall from top to bottom, no? So as you can see here, this is an example, and it pierces the cell membrane from the outer leaflet to the inner leaflet. Kita nyo naman, no? Through and through, pasok yung integral protein natin. And what's the function of your proteins again? They function as a bridge. Inside those those integral proteins are what we call channel pores. And it is through these pores that substances such as water, ions, and other molecules can actually pass through. So ito talaga yung bridge na tinatawag. The second type of protein that we have in the cell membrane are what we call peripheral proteins. So if integral proteins can penetrate or pass through the cell membrane, peripheral proteins naman are found only on one side. Like this one, the one highlighted in green, and this one also, they're only found on the outer leaflet. While this one at the bottom is found naman in the inner leaflet lang. So when they cannot penetrate through and through, that's what we call peripheral proteins. As mentioned, we have here your carbohydrates. And what are the two important functions of your carbohydrates? Number one, communication. So carbohydrates are used as antennas by the cell in order to pass on the message from one cell to another. The second function of your carbohydrates is for cell adhesion. When we say cell adhesion, it is the capability of the cell to attach itself to another structure. So, kaya niyang kumapit sa ibang structure because of this carbohydrates. So, when we say carbohydrates, just think of two letter C's, communication and cell adhesion. Now let's talk about the different ways on how substances outside the cell can enter the cell and how substances inside the cell can exit the cell. So first we have to understand that the cell membrane has selective permeability. The meaning of selective permeability is it limits or the cell membrane only allows certain substances to enter and exit the cell. Meaning, hindi niya pinapayagan lahat ng substances na makapasok at hindi rin niya pinapayagan basta-basta makalabas ang mga substances inside the cell. So, may mga iba't ibang molecules kasi na mas marami sa loob ng cell at may mga molecules din na mas marami outside the cell. So, here is a good diagram or image of it. So, outside the cell, there are more sodium, more calcium, and more chloride. While inside the cell, there are more enzymes, more glycogen, and more potassium. Okay? So yan yung concentration nila. And what keeps it this way is the selective permeability of the cell membrane. Kasi kung ia-allow lang niya na freely mag-pass through yung mga substances na yan, papasok at palabas ng cell, hindi ma-maintain yan yung kanilang concentration. So, in homeostasis, dapat ganito. There's more sodium, calcium, and chloride outside, while there are more enzymes, glycogen, and potassium inside the cell. So, there are different ways on how substances can enter and exit the cell, and we'll discuss them one by one. The first is through diffusion. So when we say diffusion, it is the free passage of substances across the cell membrane. So there are some substances such as your oxygen and carbon dioxide that can directly pass through the cell membrane's phospholipid bilayer. As you can see here, this is one example. So these yellow structures represent, let's say, your oxygen, carbon dioxide, and other lipid-soluble molecules. Pag sinabing lipid-soluble, they're made up of lipids, no? So they can easily pass through the cell membrane without any problem. They can freely pass through. And when that happens, that's what you call diffusion. On the second method, we have membrane channels. So some substances that cannot freely pass through needs to pass through a transmembrane protein channel. So what do we call again? Those proteins that can penetrate the cell membrane from top to bottom, we call this an integral protein. Diba? So as mentioned, your integral proteins act as a bridge in order to allow substances, basically those that are non-lipid soluble, to pass through the cell membrane. Okay? So yan, ginagamit sila as bridge. The third type is through a carrier protein. So a carrier protein is also similar to an integral protein, but a carrier protein has a special characteristic which later on you will understand, okay? So right now, what I want you to know about carrier proteins is that they are similar to your membrane channels. So halos pareho lang ng itsura. Pero later on, we'll differentiate them. And lastly, we can transport substances through vesicles. So vesicles act like a vehicle. Ayan. So that you'll find it easy to remember. Vesicles act like vehicles where the substances that you wanna throw out of the cell or the substances that you wanna bring into the cell are taken by the cell wall. So this is your cell wall, remember? Kukunin niya yung substances na yan No? Kukunin niya yung substances na yan and it will bring it inside, or it can also be the other way around. Substances inside the cell will be taken by your vesicle and will be brought to the cell membrane, and then they will be released outside. So basically, pag vesicle, a part of the cell membrane invaginates and forms a vehicle through which substances can be transported. Okay? Now, all of these four passages, no, diffusion, membrane channels, carrier protein, and vesicles, they fall under two major classification. These are passive membrane transport or active membrane transport. Now, what do we mean by these two? When we say passive transport, they do not require energy. They do not require energy, and they just allow substances to pass through without spending ATP. Pero pag active, they require ATP in order to allow substances to pass through. So basically, that's the major difference between the two. Passive does not need energy. Active, ATP. It needs ATP. And remember that the choice of diffusion, carrier membrane protein, kumbaga yung pagpili kung anong gagamitin dito sa apat na to, the choice between amongst the four will depend on the size, the shape, and the charge of the substance that you want to transport. Okay? So choosing amongst the different types of transport will depend on the size, the shape, and the charge of the substance. Now, here we have the different and specific types of transport. So, for passive transport, all types of diffusion. So, when you see the word diffusion, think passive. It does not use energy. So, here we have diffusion, facilitated diffusion, and osmosis. Osmosis is also known as water diffusion. Okay? So, lahat ng diffusion, passive yan. Diffusion, facilitated diffusion, osmosis, or water diffusion. They do not need energy. They can go and pass through the cell membrane, okay, whenever they want. While your active transport naman includes active transport, secondary active transport, endocytosis, and exocytosis. So all of this in bold, we will talk about in a little while. Let's start first with diffusion. Diffusion, according to the definition, involves the movement of substances down a concentration gradient. Okay? Pag sinabi daw diffusion, sinusundan niya yung concentration gradient. What is the concentration gradient? Later we'll go into that. Before we go and discuss what the concentration gradient is, first I want you to understand that a solution is the combination of a solute and a solvent. So, what are solutes and solvent? Solutes are commonly substances that are dissolved in liquid or in gas. Solutes are substances that are dissolved. Basically, ito ay mga bagay na tinutunaw o kumakalat in liquid or in gas. And whatever dissolves the solute is called the solvent. So let's take this image as an example. So here we have the final product, which is the solution. And as we mentioned, the solution is composed of two major parts, the solute and the solvent. So the solute that we have here is salt. This is the substance that you dissolve. And whatever dissolves it is called the solvent. In this case, water. So we put salt into the water and we created a saltwater solution. That's going to be handy later on as we go through the discussion. Now, what is the concentration gradient? The concentration gradient is the difference in concentration of a solute inside a solvent. And commonly, this difference is found between 2 points. Okay, so as we can see here, we have red dots, and this is our solute. and it was placed inside a solvent. As you can see here, the dots are concentrated on this part of the container. And on the other parts of the container, please follow my pointer, on the other parts of the container, there are no solutes. So basically, the solute is concentrated here. There is high concentration of the solute here and a low concentration of solute on the other parts of the container. Because it creates a difference in the concentration of solute. Meron tayong area of high concentration and an area of low concentration. This creates a concentration gradient. Okay? When you have a solution and there is a difference in concentration inside, then there is a concentration gradient. According to our book, the concentration gradient is said to be higher or steeper when the concentration difference is large. Meaning, malaki yung concentration gradient na nabubuo pag marami rin yung malaki yung concentration difference between the two. Or let's say, you put in a lot of solutes. A lot of solutes, no? Then, malaki yung magiging difference between the concentration in this area and sa concentration ng area na to. Diba? So, malaki yung difference kasi ang dami-dami mong solutes, tas doon lang siya nakikita. So, when that is the case, it creates a higher concentration gradient. And also, the concentration gradient is said to be steeper or higher kapag yung distance between the 2 points is just small. No? Kunyari, katulad nito, this is a very small container and there is a high concentration of solutes here. Pag konti lang yung distansya nila, if there is a small distance between the area of higher concentration to area of lower concentration, then the concentration gradient is higher. What does it mean then? What this means is that diffusion happens faster when the concentration gradient is large or when the distance is small. Okay, let me explain that in a while. So basically, diffusion is the movement of solutes. Remember that. Diffusion is the movement of solutes from an area of high concentration to an area of lower concentration. Yan ang ibig sabihin ng diffusion. Again, what is that difference formed by high concentration and low concentration? The concentration gradient. Kaya if I go back to the definition, diffusion is the movement of substances down a concentration gradient. Why down? Because it starts from a higher to a lower area of concentration. Okay? Hopefully that's clear. So again, we want these substances here in red to move from this area to the areas of lower concentration and achieve equal distribution or homeostasis. Yan ang gustong mangyari ng diffusion. That's what it wants to happen. Diffusion wants to move the solutes from an area of high concentration to lower concentration in order to achieve homeostasis. Okay? So these solutes will move from an area of higher concentration to an area of lower concentration in the same solution. All right? So hopefully, it becomes clear. Now, how do we apply this bullet when we said the concentration gradient is higher, etc.? So, if there is a lot of solutes placed in one area, diffusion will happen faster because there will be more solutes that will move. Can you imagine that? If there are more solutes found in the higher area of concentration, it will take it faster. It is so much faster to distribute to the other lower areas of concentration. And diffusion will happen much faster when the solutes only have to travel a short distance. Okay? Diffusion will happen faster if it only has to travel a short distance. Okay? So let's take this as an example. We have here a beaker filled with distilled water, and then you placed a cube of salt. of course, that cube of salt will start to dissolve, making or forming an area of high salt concentration. By diffusion, what these solutes want to do is to travel from an area of high concentration to areas of low concentration. So diffusion will happen, they will move until it reaches homeostasis, or until the solutes are evenly distributed throughout the solution. And this is diffusion. Similarly, when we apply it to the cell membrane, paano natin i-apply ito? Kapag maraming substance in one side of the cell, let's say maraming sodium outside of the cell membrane, it wants to travel from an area of high concentration to lower concentration. So it wants to enter. Okay? On the other hand, pwede ring baliktad. There can be substances that are high inside, such as, let's say, enzymes. There are more enzymes inside the cell. It wants to go from an area of higher concentration to lower concentration, right? So that's the application of your diffusion. This time around, we're going to be talking about osmosis. And as mentioned earlier, osmosis is the diffusion of water. So earlier, we talked about diffusion, and it is the movement of solutes. This time around, in osmosis, it is the movement of water across a selectively permeable membrane, still following the concentration gradient from an area of higher water concentration to an area of lower water concentration. So I want you to remember that in osmosis, we are not talking about the solutes. What we're talking about in osmosis is water. So let's take this as an example. We have two containers here separated by a semi-permeable membrane. So on the left, you have solutes, which is represented by the white. And on the right, we don't have any solutes. But that's okay because the the concern here is not solute concentration, rather it's the concentration of water, which is represented by these black dots. So just by eyeballing, we can see that there is more water molecule on the container to the right compared to the compartment on the left. So following the definition of osmosis, these water molecules would want to move from an area of higher concentration to an area of lower concentration. So water will then move, no? Eventually, it will reach this stage whereby water has already gone to the other side and there is no more further movement. So, what tells us that the movement of water is over and that it has already reached homeostasis? It is through the formation of the osmotic pressure. So osmotic pressure is the force that prevents further movement of water across the cell membrane. So once enough water molecules has already moved to the area of lower concentration and there is now homeostasis between the two compartments, osmotic pressure will now form, preventing the further movement of water from and to each compartment. So here is another example. We have here a beaker of distilled water. And here we have a tube composed of, made up of a very small amount of water and a high amount of solute. So of course, by following the law of osmosis, water would want to move from an area of higher concentration. So where is there more water? It's in the beaker, to an area of lower concentration, which is inside the tube. So the tube was submerged inside the beaker, and obviously water will enter the semi-permeable membrane. So as you can see, this is the amount of solution in the tube initially, but then it rose to this level. So what happened? Water molecules found here in the beaker started to move inside the tube. Kasi they want to go from an area of higher concentration to an area of lower concentration. So, kasi, di ba, mataas yung solutes dyan. There's so many solutes here. What it wants to happen is to balance out yung water level, no? So ano mangyayari dun sa very concentrated solute? Eventually, madadilute siya, 'di ba? Madadilute yung solution kasi pumapasok yung tubig. Until such time na no more water can enter because it has already reached equilibrium or homeostasis with the water in the beaker. So, what was formed and what stopped the further movement? It's osmotic pressure. Now, let's apply our knowledge of osmosis and osmotic pressure to the test. So again, osmotic pressure, the formation of the osmotic pressure depends on the difference of solution concentrations inside and outside the two compartments. In this case, we'll use a cell and a solution. So we will put a cell inside a solution of different concentrations of water and solute. And we'll compare the reaction, will compare the result of these solutions to the cell. So there we have three types of solutions. These are isotonic, hypertonic, and hypotonic. So wag malilito ha, I want you to listen carefully. When we say isotonic, iso means same, while tonic means strength. Ang pinag-uusapan dito, when we say tonicity, it refers to solute concentration. So when we say hypertonic, mataas ang solute. Hyper meaning more, tonic, more solutes. Ganun yung ibig niyan sabihin. And in hypotonic, hypo means less, tonicity means solute, di ba? So less solute in this solution. So let's start with isotonic. Iso means same, meaning there is equal amount of water and solute. Equal amount of solvent and solute. Okay? Equal amount. Pag sinabi nating hypertonic, there are more solutes compared to solvent. There are more solutes compared to water. And when we say hypotonic, there is less solute compared to water. Okay? Now, again ha, yung mga pangalan ng solution, it refers to solute concentration. But we are talking about osmosis. So out of the three, saan may pinakamaraming water? Hypotonic. Because there is less solute. Solute is represented by the yellow. And there are more water molecules represented by the blue here. So there is more water in hypotonic and less water in hypertonic. Okay? Tandaan, yung hyper tsaka hypo ang tinutukoy niya solute. Pero when we talk about water, opposite na siya. In hypertonic, there is less water. In hypotonic, there is more water. Okay. That will come very importantly when we discuss them one by one. Ayan. So, anong gagawin natin ulit? We will put a cell inside each of this solution and we'll try to see how water will move, no? From the solution to the cell or cell to the solution. Let's see. So, first, we have a hypotonic solution. Again, hypo means less. Tonic means solute. So, less solute here. So, it is an area, it is a solution with a low concentration of solute but a high concentration of water. Okay? So, we put a cell inside the hypotonic solution. So, what has more water? The cell or the solution? Obviously, it is the solution kasi there is higher concentration of water. Okay, there is higher concentration of water. So what does water want to do? The water wants to move from an area of higher concentration to lower concentration. So water will move from the solution, it will move inside the cell. So water moves by osmosis into the cell, causing the swell to get bigger, or to properly say, it causes the cell to swell. Because there is so much water entering the cell. No, pasok ng pasok ang water. The cell will swell until it reaches its limit. No, until it reaches its osmotic pressure, no? And it will rupture. And the rupture of cells is called lysis. Pag pumutok yung cell sa sobrang daming water niya inside, we call it cell lysis. So, yun ang nangyayari in hypotonic solution. Once you put a cell in a hypotonic solution, since there is less solute and more water in the solution, water wants to move from an area of higher concentration to lower. So, the water will move from the solution into the inside of the cell. The cell will then swell until it reaches its rupture point and it will manifest cell lysis. Okay? Next, we have isotonic. So from the definition earlier, iso means same. So there is the same solute concentration inside and outside the cell. Okay? So what will happen? There will be a balance movement. Kung may water na papasok sa cell, lalabas din yung water na yan. Kasi equal nga lang yung amount nila. So pag nabawasan yung isa, maglalabas yung isa. No? So in isotonic solutions, the cells will neither shrink nor swell. It will remain the same. No net loss or gain. Next, we have a hypertonic solution. So what does hyper mean? It means more or it means high. So ano yung mataas sa kanya? It has a high solute concentration. But in terms of water, there is lower water concentration. And then we put the cell inside the hypertonic solution. So saan ngayon ang may more water concentration? Yung cell. The cell has more water. So what will water do? Water will move by osmosis from the cell towards the solution. So water will get out of the cell. So what will happen to the cell? It will shrink. And cell shrinkage is called crenation. It will shrink and shrink as water goes out continuously until it becomes so shriveled that it cannot give water anymore. Okay? So putting the three together, this is what happens. We have here an isotonic solution, a hypotonic solution, and a hypertonic solution. So in the isotonic solution, when a red blood cell is placed inside the isotonic water, there will be a balance of movement. No? Water just moves in and out of the cell in similar rate. So there will be no affectation to the cell. It will remain normal. But if we place a cell inside a hypotonic solution, hypotonic means low concentration of solutes but high concentration of water. So since there is higher concentration in the solution, it wants to go from the solution towards the inside of the cell. So the cell will get swell, it will get bigger until it lyses or until it ruptures. The opposite happens in the hypertonic solution. So in the hypertonic solution, there is more concentration of solutes but lesser water, meaning there is more water inside the cell. So that's the area of higher concentration and it wants to move to an area of lower concentration. So lalabas yung tubig. Labas ng labas yung tubig towards the solution until the cell shrinks and shrinks and shrivels. And that's what we call crenation. Okay? Now that we're done with diffusion, and osmosis, we are now going to be talking about the second type of transport, which is carrier-mediated transport mechanisms. And under this type of mechanism, there are two subtypes, which are your membrane proteins and your carrier proteins. Ayan. So, in diffusion, as mentioned, the lipid-soluble substances such as oxygen and carbon dioxide can just freely pass through the phospholipid bilayer. But how about the non-lipid soluble, no? Such as your water-soluble substances? They can pass through, enter, or exit the cell membrane by passing through cell membrane channels or carrier proteins or carrier molecules. So let's try to figure out the difference between the two. So when we use membrane channels, that is called facilitated diffusion. Diba? So facilitated diffusion falls under carrier-mediated transport. That's the first, this is the first type under carrier-mediated transport. And When we say facilitated diffusion, this is the movement of substances across the cell membrane from an area of higher concentration to lower concentration. Basically, since it is still a diffusion, it still follows the concentration gradient. Okay? Thus, since it is a diffusion, it does not require ATP or passive transport pa rin siya. So, what happens in facilitated diffusion? Your integral proteins or your membrane channels forms a bridge in the middle which allows the passage of your non-lipid soluble molecules and ions in and out of the cell. Simply, ganun lang yung nangyayari. Again, from an area of higher concentration to an area of lower concentration. There are two types of your membrane channels. First is the leak channels, and second are your gated channels. When we say leak channels, they are constantly open and they constantly allow ions to pass through. While your gated channels may gate. So, pag may gate, it limits the movement of ions depending on when they are opened and when they are closed. So, let's look at it. This is a membrane channel. A membrane channel at integral protein ay iisa. They refer to one and the same thing. Okay? So, a leak channel is opened on top, opened at the bottom. It's always open. Okay? It's always open and it always allows substances to pass through. While your gated channels depend on whether it's open or closed. So, nalilimit yung pwedeng pumasok. So, if it's closed, nothing can enter. But if it's open, then substances can enter. Okay? That's the meaning of it. The second type of carrier-mediated transport are carrier molecules or carrier proteins. Okay? So these carrier molecules are still proteins. They're still proteins. But what's special about your carrier molecules is that they exhibit specificity. What do we mean by that? Ang bawat isang carrier molecule ay may specific molecule lang na inaalaw niyang dumaan. Each carrier molecule will only allow a specific molecule to pass through. That's the first important information about carrier molecules. The second important information about your carrier molecules is that they undergo conformational change. Ano ibig sabihin nun? What's the meaning of that? It means they undergo shape transformation. Here, so this is a carrier molecule. It still is an integral protein. But the first difference, it exhibits specificity. So if you can see here, the carrier molecule has a shape. And it will only allow substances to pass through if the shape of the substance fits the shape of the carrier molecule. Kuha ninyo, papayagan lang niya dumaan yung mga substances na mag-fit doon sa kanyang shape. So, as you can see here, that yun yung una. That's the meaning of specificity. The second important information about carrier molecules is that they undergo conformational change or shape transformation. As you can see here, the top part of the carrier molecule is opened while the bottom part is closed. But once a substance exhibits specificity or pag kumabit na yung substance, magbabago yung shape ng molecule. The top part now becomes closed while the bottom part opens up. Okay? And it allows the substance. This is different from your gated channels. Because in gated channels, there is no change in shape. It simply opens and closes the gates. But in carrier molecules, it changes its appearance. Okay? All right. Next, we have active transport. So, active transport is still under carrier-mediated process. No? Ito yung pangatlo. So, ang kakaiba kay active transport, it requires ATP. And look at this, the movement of substances is from regions or areas of lower concentration to areas of higher concentration. So, it's the opposite of the concentration gradient. Thus, the movement is against a concentration gradient. So what happens in active transport, no? In active transport, it accumulates the necessary substances on one side of the cell membrane at concentrations that are greater than those on the other side. So remember earlier I showed you what substances should be abundant inside and outside the cell. What keeps those substances pala abundant in their respective areas is active transport. Just like your sodium and potassium. Let's see. So, the sodium-potassium pump is one of the major examples of active transport. So, what does this pump do? It moves sodium out of the cells and moves potassium into the cells. So, in order for you to remember the normal concentration, it's piso. Potassium is in, sodium is out. And what keeps sodium more abundant outside and what keeps potassium more abundant inside is the sodium potassium pump, which is an active transport. All right? So let's look at it. Ayan. As mentioned, your active transport is still a carrier-mediated process and it uses ATP. So what it does is it brings out three ions of sodium in exchange of 2 ions of potassium. Okay? So 3 sodium out, 2 potassium in. Thus, making sodium more abundant outside and making potassium more abundant inside. Now, there is such a thing called secondary active transport. So, because of that movement created by your active transport, there will be abundant sodium inside and abundant potassium inside. So, the secondary active transport uses the energy provided by the concentration gradient that was established by the active transport, di ba? So, sa madaling sabi, ginagamit ng secondary active transport yung concentration gradient na create ng active transport mo. 'Di ba? During the active transport, there is more sodium outside and more potassium inside. So the active transport results into a new concentration gradient, 'di ba? So dahil there is more concentration of sodium outside, it wants to go in naman. It wants to go in naman, 'di ba? So no additional energy is required by a secondary active transport. So let's see. Ayan. So this is your active transport. 3 sodiums out, 2 potassiums in. And then this is your secondary active transport. Because there is now too many or too abundant sodiums in outside, ang tendency naman niya is it wants to go in. No, it wants to go in kasi nga from area of higher concentration to lower. So gusto niyang pumasok. At pagpasok ng sodium, magdadamay siya ng ibang substance. It will bring with it another substance. And that is your secondary active transport. This is just an effect of your primary transport. And there are two types of secondary active transport. Your co-transport and counter-transport. When we say co-transport, the diffusing substance moves in the same direction as the initial active transported substance. So in this image, the initial transported active substance is sodium, 'di ba? So sodium wants to go in and glucose wants also to go in. So since they are of the same direction, it's called co-transport. And the opposite is counter-transport. When the diffusing substance moves in the opposite direction as the initial one. So let's say that sodium wants to get in and glucose wants to get out. So since they are of different directions, then that is a counter transport. I think this is one of the last few types of transport. We have endocytosis. So when we say endocytosis, it is a process that brings materials into the cell, and it uses vesicles. Diba kanina we mentioned already what vesicles are. These are vehicles created from the cell membrane. Okay? So when we say endocytosis, you want to bring materials into the cell. Okay? And then you'll keep them in vesicles. So what happens? It begins with a receptor-mediated endocytosis. So what this means is that the substance will need to bind with a receptor found on the cell membrane in order for it to be transported. Let's look at it. Ayan. So these purple circles are the substances you want to bring into the cell. But they cannot just enter. They need to attach to a receptor found on the cell membrane. After they attach, that part of the cell membrane will go deep. It will start to burrow in, lulubog siya in other terms, until it creates a new a circle, it creates a vesicle, and it will now be brought to its target destination. So, if the material that was taken in is a solid, then you call that phagocytosis or cell eating. But when the material that is taken in is a liquid, then you call it pinocytosis, also known as cell drinking. So, these substances will again attach to your receptor molecule. The cell membrane will burrow in or lulubog siya until humiwalay siya from the cell membrane. And then, it will be now called a vesicle. If the substance is solid, it's called phagocytosis. If the substance is liquid, it's called pinocytosis. So if substances can be brought in through endocytosis, substances can also be brought out through exocytosis. So in exocytosis, the vesicles are specifically called secretory vesicles. Yan ang tawag sa mga vesicles na nagre-release ng substances to the outside of the cell. Okay? Again, exocytosis is for the release of substances from inside to the outside. So what happens is that the secretory vesicles will move towards the cell membrane, will fuse with the cell membrane, allowing the release of the materials. So let's see. Ayan. So here we have a vesicle containing the different substances you want to release. So what the vesicle will do, it will attach itself to the cell membrane. It will fuse until an opening is created like this one. Once an opening is created, the vesicle will continue to push the substances outside until they are completely released, such as seen in this image. And this is what we call exocytosis. Now, when does the body or when does cells use exocytosis? It uses exocytosis when it wants to secrete digestive enzymes. So, yung mga enzymes na ginagamit para makapag-digest tayo, they are created inside the cell, di ba? Function ng cell yan. Creation of protein, lipids, other organic molecules. So, pagka-create niya nun, it will release it towards the outside so that the enzymes can start to digest the food that we eat. Okay? So, that's what happens. Okay? So, we'll end the lecture here and then I'll be giving another video recording of the second part of our cell anatomy and cell physiology lecture. Please stay tuned for that. Thank you guys and see you.